Hello, everybody. My name is Assaf Zaritsky, and I'm happy that you could join us to the second part of the American Society of Cell Biology Data Science webinar series. Okay, so this is the second session. You can find the first part. You can find it was recorded, and you can find it in the ASCB site. Today, we're going to focus on what is deep learning and how to use it in cell biology. Then in July 9th, we're going to have a session about collaborations, where we're going to have a tandem talk of pairs that collaborate for a long time, and a computational talk collaborating with experimentalists, and experimentalists collaborating with a computational scientist. And then the last session is going to be about how scientists see the future of data science in cell biology. The session today is going to be 3 20-minute talks without questions. And then we're going to have a panel discussion that includes the questions to the talks and other issues. Feel free to type in your questions while the talks are ongoing. Before we start, I have two administrative issues, or promotional, better saying. One, I want to encourage you, if you have cool quantitative cell biology papers, to submit them to the American Society of Cell Biology Society Journal. Why? First, it's not-for-profit. All the profits goes back to us. ACB members get a guaranteed peer review, and it's fast and fair and constructive, so really try it, and you're welcome to approach me or one of the other editors and consult if you'd like. And last, Meghan and I are also organizing a subgroup for the American Society of Cell Biology. So please first register to the meeting, and then we're happy if you can submit to us your cool projects. And with that, I'll turn to Meghan, and we'll start the session.
Right, so first up we have Beth Cimini, right? She's the Associate Director for Bioimage Analysis at the Broad Institute, which is a joint institute run by Harvard and MIT. And her group makes the well-loved CellProfiler image analysis software, as well as collaborates with experimental groups to do great science. Well, that's it. Take it away.
All right. Hello, everybody. Thank you so much for joining today, and thank you so much, Assaf and Meghan, for having me. I'm going to talk a little bit today about what is deep learning and why might you want to do it. I expect we have people here from a range of different scientific and computational backgrounds, and we want to make sure that everybody's on the same page before we dig in further. So at least a couple of you may have found yourself here, saying you're not really sure what deep learning is. You know that it's everywhere. You know that it's being used in a lot of different tools at this point and that it's powerful, but what exactly is deep learning, and what are the advantages of using it, and what are the sort of potential things you need to think about when using it? So deep learning can help us get data from bioimages in a lot of different ways. We can do things like we can predict staining that we didn't actually do. We can sort of do a virtual staining. We can predict the outcomes of different assays from images. And the one that's most common that you've probably done if you've done much image analysis, we can use deep learning for doing cell segmentation. And all of these are not hypothetical. These are all real things. Some of these are from our group, and some of these are just from groups that we're big fans of. But there's a tremendous amount that we can do if we leverage the power of deep learning. And so just to sort of start with, what are we talking about when we're talking about deep learning? So deep learning is a subset of machine learning, is a subset of AI. With deep learning, we're talking about machine learning algorithms that use deep neural networks in order to actually solve particular problems. But there are lots of cases where you might use what we'll sometimes call conventional machine learning, where you're having a computer solve your problems, but not using these specific kinds of neural networks. And deep learning is basically just saying, I have an input and I want to be able to predict an output. I would like to be able to say, what is in this picture? What's in this picture is a cat. And so I'm going to give you some slightly simplified, but more or less correct definitions, and then tell you a little bit more about some tools for doing it and ways that doing it is hard and ways that doing it is sort of hopefully a little easier. So we're talking about deep neural networks. The idea is essentially we have inputs and outputs like we saw before, and we've grouped things into layers. Each one of these things is typically called a neuron, and a neuron is typically just a filter. I'm going to show you an example in just a minute of what this looks like to make this a little bit less abstract. But the idea is we connect all of the nodes in our network, and we adjust the strength of the connections so that the output comes out the correct way. And so this allows us to create complex relationships between the data that we have and the answers that we want. But because we're trying to do this in a way where we don't understand exactly how to do that, we tend to just sort of keep stirring the pile until the data looks right. And here's what stirring the pile actually looks like. So this is playground.tensorflow.org. I definitely recommend playing with the site. It's a great way to get sort of a understanding and an intuition about deep learning. Here we have neurons that are all of these different filters, and we adjust the strength of the connections and essentially say each time we sort of make an individual connection stronger or weaker, does it get a little bit better or does it get a little bit worse? Are we better at predicting this idea of a spiral? So let me run it back and, oh no, can I play it again? Okay, apparently I cannot play it again. Well, I can play it again if I restart the slideshow. So here you can see that the connections all start at the same strength and the same width, and the width here is symbolizing the strength of the connection. And over time, as the thing tries to learn the idea of a spiral, what it does is it actually goes through and says some connections are really helpful for this, some connections are not really helpful for this. And it eventually sort of comes up with the idea of a spiral. We haven't let it go very far here. You saw this only ran for a few seconds, but the longer we let this run, the more it will try to get the idea of what a spiral is by just adjusting the strength of these connections and saying better or worse, better or worse, better or worse each time. And there's a lot of different kinds of neural networks. I'm not going to go into any detail at all about the specific kinds of neural network that these are. I've just sort of highlighted some of the ones that are typically very common. So if you're talking to your computer scientist friends, you have some things that you can ask about. A lot of things, especially in segmentation, are based on what's called the UNet architecture, but we're seeing also lots of things coming up these days with things like transformer models and variational autoencoders. But in general, all of these things are around this idea of strengthening or weakening connections and saying, did my sort of prediction of a particular output get better or did it get worse? CNNs or convolutional neural networks are one of the most common things for learning from images. What exactly is state of the art goes back and forth, depending on whose papers you believe and whose papers you're reading this week. But it's still, I would say, the most common and arguably is state of the art. So the UNet architecture looks like this, where we start with sort of big uncompressed data. We sort of compress our data into different shapes as we go through and then uncompress it at the end. And it turns out this is an extremely powerful way to actually understand things about what is going on in the images, because we sort of compress into this smaller space where we've said, here's a whole image worth of information, and now we're condensing it into just sometimes as few as a couple hundred different numbers in what we call our latent space, which is the sort of place in the middle of the network. And it's important to talk about latent space, and it's important to talk about this idea of what actually is a neural network learning, because it's really sort of hard to understand from a biological point of view what a neural network actually is. If we go back and we look at sort of this idea of a UNet and the fact that we have filters and we have data that is sort of being compressed down into a particular shape, if I want to know what particular numbers is the difference between sort of understanding that this is a cat or thinking that this is a dog, if I go into different layers of my network and I ask essentially what the network thinks that layer is about or what that level of information encodes, it can be really difficult to understand. It can be hard to figure out what has my network learned. And so you lose interpretability a lot of times with deep learning, although there's certainly a lot of people working in the field of explainable AI and explainable deep learning and working to sort of crack open that black box. If you've decided that you want to train a neural network, you need a lot of data, and we need some sort of annotated data. So we need to know essentially what are the right answers for any problem that we're trying to solve, especially if we're doing so-called supervised deep learning. And I'm going to talk about unsupervised in just a minute. So for example, if you want to train a segmentation network, you typically need to know where are the objects that I want to find. And so if I want to find cells, where are the cells I need to find? And you typically have to go through and create all of this, what's so-called ground truth that says exactly what is the right answer that the computer is supposed to be learning to predict at the end. And so what you'll typically do is you'll go through, you'll put in a sample data set, you give it the correct answer, you see how well it predicts it, and then again, you go through this better or worse process that we showed in the playground. But the problem is that I said we're fitting very complex curves with a neural network. We can fit essentially any relationship we want between our input images and our output answers. And we typically have finite amounts of training data that we put in, especially when you're talking about something like segmentation, where hand drawing in all of those cells, I can say from experience, takes dozens or hundreds of or thousands of hours to get enough data to actually train a neural network completely by yourself. And if you don't actually create enough data, you can actually end up with your network so-called overfitting. And so that means that essentially what the network has done has memorized the data that's there and not actually learned how to predict anything. It's just memorized all of the data that you gave it. So here's what this looks like in practice, where we've overfit that playground model that I showed you earlier, where we're trying to learn the shape of a spiral, where we only gave the network a very small subset of the total spiral that we want to learn. And so the network has done a really bad job of figuring out which parts of this image should be orange and which parts of this image should be blue. But it's understandable that this network did a bad job because it never saw anything up here in the orange, where we know it should be orange based on the whole data set, but it simply never saw an example of what that should be. So it's really important when you're doing deep learning to be able to not overfit and to be able to create training data that actually covers the whole subset of the data or the whole superset of the data that you want to be able to use this network to predict, whether that's a classification or a segmentation or a denoising, anything else. And because, again, it's very hard to create this ground truth, overfitting is something that happens a lot. We all did the work for for pictures and natural images, which is photography, for creating ground truth. We all went on Facebook and we tagged people in objects, and big companies like Google and Facebook spent a lot of money creating big data sets with literally millions of images where we know what all of the objects in them are. We don't tend to have that in biology. So again, unless we make it ourselves, and that can take months or years. But so we can take advantage of the ability that that we got from these bigger data sets, though, to sort of try and leverage and work on top of them. And that will help us reduce the amount of overfitting by having us have a network that already understands a little bit about images anyway. And so how this works is that rather than starting all of our weights at random, again, if you think back to the spiral we looked at before, where all of the connections were equal, we can train a network to understand something about where are objects. We can say where are planes and bikes and cats and dogs. The network isn't then magically going to know how to find cells, because cells don't look like airplanes or bikes or cats or dogs, but it at least understands things like foreground and background and textures and things that might really be valuable for finding cells, even if cells don't look like cats or dogs. And so what this allows us to do is essentially hone in on the problem. If you think about the fact that we're sort of in this huge landscape of possible deep learning models, in theory, all possible combinations of neurons. You might be, if you only have the ability to sort of look within a really small range because you really have a small amount of training data, you might want to make sure that if you're trying to find a particular place, that you're starting on the right part of the map. So you're not starting over here, where you're just completely in the wrong spot, but that actually, if you want to find the highest peak, that you can find it, you're already on the right mountain and just have to go a little bit further to find the highest peak in your landscape. So being able to start as close as possible allows you to have the smallest possible set of training data, which is the smallest amount of manual work that you need to do. And so one great piece of software that if you haven't tried, I definitely recommend that you do is CellPose, especially versions 2 and 3, and is a great example of the ability to sort of use pre-training to actually make it a lot easier to do deep learning. And so this is from the CellPose 2 paper from a couple of years ago, where they say if they are trying to figure out how to find these bright field images of cells, if they start from scratch without sort of pre-initializing the weights, and just sort of have everything start from random, it takes a lot of images, 6,000 crops of these cells that somebody had to go through and annotate all of the right answers by hand in order to actually reach human agreement accuracy. If you start from something that's already been trained, you can actually get there much, much faster, and you can get to a higher level of performance. And in fact, if what you do is you sort of have a human go through and you give them feedback every single image of, how good of a job it's doing, in 1 or 2 images of annotation, you can actually reach sort of within human agreement. And so these sorts of human in the loop training methods that are coming online, and again, CellPose is a great example of how to do it, is making it easier for you to not have to spend 3 months making training data and then sit down and train your network. You literally only make as much training data as you need, and that'll help you get going with deep learning. And I said before that it was helping reach human agreement levels. One thing I always want to caution folks with when they're trying to do particularly segmentation tasks, within human agreement is actually very bad for a lot of segmentation tasks. You may have noticed here that within human agreement on this is actually a sort of average precision, or the amount that the two different humans agree on where the exact boundaries are, is actually only about 0.7. So you're never going to get a network that shows up as 100% agreement with ground truth, and if you have, it means you probably overfitted it to one person's idea of cells. And so this is a preprint that we recently put out showing that if you give this a person the same images rotated 90 degrees and you have them annotate them in each orientation, they only actually agree about 65%, which is about as much as they agree with a computer model. The computer is much more reliable and reproducible. So it's a great example of sort of why you need to use deep learning, but also how to think about where deep learning adds value, because again, it's more reproducible. Because all of this is very costly, there is this whole field of unsupervised learning, which I'll mention for just a couple slides, where we try to figure out how we can do clever ways to sort of tell the network what we want it to learn without us having to sit down and spend weeks or months. We can do this by coming up with a pretext task. So we can say, all right, here, what is the thing that we can teach the network that's almost exactly what we want it to do? And so those of you who've used ChatGPT or other large language models, the idea that those are trained on, many of them are things like auxiliary tasks, like dropping out a particular word, or saying, does this sentence come after this other sentence? And in biology, we can come up with similar ideas for sort of unsupervised learning. So if you have access to lots of data and not much ground truth, these are the sorts of methods that you can do. And if you think about ways you can bring in truth, such as, are these both from the same image or not, you can come up with clever ways to train neural networks without so much data. Just want to briefly mention metadata is a thing that if you want those of us who are computer scientists in the field to keep making you great deep learning models, put up publicly annotated data and put it up with consistent annotations. It's really hard to come up with neural networks that solve a lot of problems if we don't understand what's going on in the images. And it's a really important part of making your data fair, findable, accessible, interoperable, and reproducible. Deep learning is typically difficult to access for non-computational users right now. When we ask people how comfortable they are doing image analysis, and how much time they spend doing image analysis and how comfortable they are developing new computational skills, people who tend to work in the wet lab tend to be less computational. And that tends to mean that the people who are training deep learning models are not necessarily biologists and not necessarily experts in the phenotype. And so the problem, because it's difficult for these people, because these people are not computationally comfortable, it means we want to make deep learning easy to use, which it currently typically is not. Installation is actually a huge problem with deep learning, as those of you who may have tried to start using it may have come across. And so I just quickly want to mention, as I'm wrapping up here, I know I'm just about out of time, that we as a field are trying to make web tools, and I think you're going to see some later from things like the Bioimage Model Zoo, to make it so that people can access deep learning on the web and can actually do deep learning without having to do installation, because we, the developers, have installed things for you. So if you've tried to get started with deep learning and you haven't actually been able to do it yet, there are good tools that exist and more tools that are coming that are going to help you do this. There are things like Galaxy, things like image.j.js, things like zero-cost deep learning for microscopy, as well as a tool that we're making called Piximi, which allows you to do deep learning classification and segmentation, as well as annotating images and measuring them all in the browser. I don't know what. And so before I turn over, I'm just going to sort of say, we know that we are trying to make things that make deep learning easier, but we need to hear from you how we're doing. We need to hear from you what actually is needed. And so I definitely recommend, if you remember nothing else from this talk, to remember forum.image.sc. It is an open forum where you can get experts from more than 60 different pieces of software, and you can ask them questions, whether it's about deep learning or anything else, or you can just post a picture and say, this is my picture, this is my question, please help me. That is funded by the Center for Open Bioimage Analysis. And I just want to wrap up and say thank you to the folks in my lab who make awesome deep learning tools like Piximi, and as well as some educational materials that you can ask me more about when we get to the panel in just a bit. Thanks so much.
Okay, well, thank you, Beth. Like Beth said, we're going to have a panel at the end of the talk, so please ask questions then. All right, so next up we have Anna Klemm. She runs the Bioimage Informatics unit at the SciLife lab in Sweden, right? And her unit collaborates with wide-ranging projects, ranging from the small scale to giant team efforts, right? Go for it.
Great, thanks a lot. Thanks for organizing. Thanks for coming. And yes, indeed, I want to share a bit what we are doing in terms of deep learning, because, okay, I'll share my slides first. So the idea, what I thought to show is projects that we meet in our support unit, where and how people are using deep learning, and in which steps, how difficult it is, which tools are around. So that was the idea what I brought today. And yeah, so indeed we are an entire unit providing support in image analysis. And what we see are projects from researchers all over Sweden. So meaning we see quite a variety of problems and data types, and then we get experience in where which tools we can use in these projects. And I think first thing that I want to highlight is that we still use a lot of classical image analysis, but then we use more and more deep learning based tools also. So we have a lot of what we see in Sweden basically is a lot of these spatial biology, so tissue data, tumor data, and I will tell you in a few slides that especially there you can use these pre-trained networks that Beth was already mentioning in her introduction. Good. So how does this typically look like when people come to us? So it does not really happen that people come and say, this is the data we thought about using deep learning. I mean, sometimes happens, but usually that's not the case. But what does happen is that researchers come to us and they have their data set and they have problems analyzing them. Maybe they have already experience using a CellProfiler pipeline and or some Fiji plugin and so on. And they get stuck, and then, or additionally, they maybe even earlier want to know how to automate their analysis. And then we typically still look into, okay, can classical image analysis be used? And try to build some pipelines there. But then also, we also try to point them maybe to like machine learning tools. So basically a pixel classifier, as you might know from Elastic or inside QPath. So there you already train and annotate data, but then it uses more classical filters and to make predictions on your data. But then, okay, once this is clear that it's not possible, then of course, or we kind of then like to use, or in general, we like to use also these pre-trained models, and then also the step of, okay, do we want to train and retrain a model or even build an entire new architecture for them? And then there it depends a lot on what is the data, what are the annotations, what experience do people have, and with infrastructure is also available. And I indeed wanted to start with an example where we did not use deep learning, but just machine learning. And so I will show a few examples on how we deal with them. And then also I give some practical tips in the end. So for this first example, what there was is like multi-channel images where each channel reflected a marker. And then the marker was about, so the entire story was about spermatogenesis in a group here in Uppsala. And like every marker is pointing to a phase. And then they additionally had one protein where they are interested in where it is located in which phase. And then typically what happens is when the markers are very clear and good signals, so classical image analysis works well. And then for the markers that are, for the protein that is unknown, or for where the function is unknown, there typically we expect a more higher background maybe, and like more variety in how the signal looks like. And then it becomes more tricky if you want to have an entire workflow handling that. And then in this case, for example, we recommended to use the pixel classification in the tool Elastic. And then so they were marking positive and negative signals, and that was enough for this project. So they had to mark a few of these images, but they did not need to build now a huge annotation base and then go into deep learning. Good. And okay, and that was kind of to show again, you can do a lot with classical image analysis and machine learning. But then, of course, especially pre-trained models are good to know. And then, of course, you also sometimes want to retrain. And starting with pre-trained models, I think by now, STARDIST is kind of very well known. But I think I still want to highlight that it outperforms in many cases classical image analysis by far. And especially in tissues that are so we want to, all of this is to segment nuclei, and in tissues where you have very dense nuclei, and which, like in this example here, where you can see like nuclei of different sizes with rough texture, then what you would get with a classical auto threshold is usually not as good as what you would get with the STARDIST pre-trained model. And the nice thing is also that here there was a lot of work put into making this available to biologists, so you can just in Fiji try it out, and then also check what the parameters are doing in Fiji. And if you're familiar with Fiji, you have this all in this environment that you are familiar with, like with the ROI manager and so on. And quite maybe slightly less easy, but still very easy. It is to run, for example, in QPath. So in this, if you have like these huge digital pathology images, then you can run it very well also there. And where do we use this? So of course we use it like in difficult images, where so again here is like a colored images of cells. And then here the segmentation would be rather tricky with classical images analysis and work very well with Stardust. And then we also like to combine it with like, yeah, segmentation of other structures, measuring the distance of nuclei to these other structures and so on. Yeah, closing this. And then, yeah, you also heard about CellPros. So again, what I want to point out here is this is basically another pre-trained model. You can also use it for just segmenting nuclei, but also for entire cytoplasm. And I think what I want to highlight here is, okay, how do you choose what is more appropriate for your example? And for this, it's always good to just try it out. And luckily, they have a drag and drop thing in their website. It is only the old CellPros version, but you can already get a good impression. But then also you can look at the example data that they give to give an impression of whether it could work for your data or not. Good. And yeah, I think, yeah, here there was okay. Just to mention, so there are really a bunch of pre-trained models for various tasks. And one also quite famous one is this deep lab cut. So this is more for, or this is for pose estimation, meaning like if you have a mouse walking around, you want to track where the different, like how the movement happens, then you can do that. I tried this, for example, out with the pre-trained human model on myself on this rowing machine, and this is indeed very like simple to do. There's much more than these examples. I guess these ones were the most, well, one or for segmentation, the most famous ones maybe. But it is good to be aware that there are resources where you can check what other pre-trained models are around and where you can also test them. And here I want to really highlight the bioimage model. So, and I guess Florian will talk more about this. But so I opened here the part where you can really see kind of the applications that you can find models for. And then some are more for segmentation, some are more for denoising your data. And then you can see also different modalities that you can use the models on. Good. So let's go back to our kind of typical requests. So the next step is, okay, so I think this running pre-trained models, this is really day-to-day business by now, and some work easier than others, as Beth already mentioned a bit. And then sometimes you see that the pre-trained model does not work perfectly on your data, so you wish to retrain it. And also there depends a lot on the framework of how easy that is. In some ways it's quite easy, in others it's quite difficult. And I picked two examples here where it's rather easy. So in CellPose, they have this nice GUI, so you can run the normal CellPose, the classic CellPose model, and then start correcting it and then rerun, given that you have a good compute infrastructure behind. And then also already mentioned by Beth was the zero cost DL for MIG, which the aim is to help you like avoiding to install everything by yourself, which is difficult, but also provide compute infrastructure to be able to run this. Okay, and then how does this work look like again in our unit? So what we see is we try to help people, of course, running existing pipelines also, just, yeah. So what you would see here is an H&E image where they had many of these tissue slices and they wanted to segment a certain structure, the salivary glands, and then they had highlighted or annotated them quite extensively in QPath, and then with this no-code segmentation pipeline that is out there, we could help them to retrain, to train a network, to then load the outlines again into QPath, and then you can go several cycles and get a perfect model for the data. And again, a different example how this looks like in our unit. So if people then really, I mean, of course, in such a support unit like ours, or like there, then what you can do is to really help building a model for them. So in this case, it was about looking at these venules here and classifying how, like, whether they are dilated or not. And then again, what the biologists brought were very nice, really good image data, and quite a lot of, meaning 12,000 annotated of venules, and also like annotated, like outlined and classified. And then this is a data, of course, that you then can use for deep learning. And I think in this case, for example, what we did was building our own interface to run this, because we were, like Christoph from the facility, he was combining it then with other classical image analysis pathways. I think, yeah, nowadays it's, I guess, would be more ideal to use what is already around. And yeah, good. This got a bit cut. But I think the last minutes, I would like to spend a bit on how difficult is it for you to get started. And I guess in general, it depends a lot on the tool. But just to get some impression, I tried to make this table here and saying where could you also, in which way could a bioimage analyst help you? And maybe I try to just go out of the screen sharing to get the entire a slideshow to get the entire table. So what I told you is, okay, you can run these pre-trained models. This, depending on the model, can be, is in some, depending on how much work also people put into making this available, this can be extremely easy and satisfying to do. And then maybe the main job of a bioimage analyst would be then to find the right model and to help you, to point you, for example, to the bioimage model zoo if people don't know it, and choose the right model and then help you to run it. And then if you go then one step further, then often it happens that you need to create your own annotations. And then, of course, what you need is a smooth tool to draw these outlines. And as Beth also already mentioned, this can be a lot of work, so you want to make this as convenient as possible. And then here, what you could get help with is in the choice of tool you could use for that, and then also how to export this in a format that someone can work with that. And sometimes you can use like a magic wand or something that at least helps you somehow to create these annotations. And then we already talked about, yeah, like retraining a model or even trying to build your own. And then, of course, what you need is infrastructure to run this. You need the knowledge about, yeah, how to do this. Or if you build your own, of course, like about the architecture, hyperparameters, and so on. And there again, of course, then it's really useful, I guess, to discuss this with an expert who can provide or point you to the right compute infrastructure, but also consult you, like what choices can you do if you want to go there, and how to evaluate all of this. Good, and then I go back to the slideshow. And yeah, so since annotation is indeed important, it can be done in tools. So yeah, in tool like in QPath, for example, I often recommend to use because it's quite smooth. You can export it easily if you have a 2D data set where you want to annotate. And but also, like in this one example that I showed, they were annotating in Fiji and had saved in the ROI manager. Again, they were very used to that, so it's good. And I also recommend already CellProfiler and using this identify objects manually. So it depends also a bit, not only, of course, it depends on the data, but also it depends on what are people comfortable with. And yeah, in many of these, like in ZPros, for example, as I already mentioned, you can then run the standard model and correct it. And we have also seen that if you have several annotators, that is a good thing to do because you get a feeling of the agreement that you have between these annotators already. And then kind of what do you expect from your deep learning model then in this aspect. And yeah, I put this very rough, but it's good to have balanced classes. So let's say you have like annotations of different classes, like these dilated vessels that we had, then often it's good if you have similar amount of these annotations, but it depends also. And I think this, I jumped, this was basically again to highlight, yeah, if you have several experts, then they don't agree. And okay, so I showed you an expert can help you, but where do you get an expert? So there are local bioimage analysts that this is a job right now. So you can, if you're happy, lucky, then you have one close by. If you don't, then you can look at the Global Bioimage Analyst database, for example. So there's the link here in the corner, and there's a list of experts that also show, like, indicate which is their expertise field and whether you can contact them. And then I guess it's also good to know the country where they're located, and then you can reach out. And similarly, if let's say you don't have directly access to a bioimage analyst, then the forum was already mentioned. So there you can shout out there. There are there's of course much material online for courses. So even if the course is already over, then people like to, it's good if there's material there. And maybe also a tip is if you don't have a bioimage analyst close by, maybe at least, it's a bit more common to have someone, an expert in the computing resources and bioinformatician. So they might also be able to help you then with running, for example, retraining a model using the resources. And with that, I want to thank the Bioimage Informatics facility at Scilab Lab here in Sweden, and for your attention.
Okay, well, thank you, Anna. Remember, we have questions at the end, right? So please stick around and ask the panel lots of questions. So next up, we have Florian Jug. Florian is a group leader of the Human Technopole in Milan, right? Not only is he and his group both an advocate and an implementer of open science, right? But they've also developed many elegant machine learning algorithms to forward biology and microscopy image analysis. Go for it.
Thanks a lot for the introduction. Can you please stop sharing the screen so that I can start, Anna? Super. I have interestingly almost nothing new to say, and I will still go over time. I'm kidding. I try to stay in time. So I prepared a few things. I have to speak about AI for Life, obviously, because otherwise I will be fired. And then I have a few other things to say. I'll come to this later. First, I thought I will quickly introduce what the lab is concerned with for a number of years now. It is things like this. I'm just flashing results, denoising of volumetric microscopy data via supervised deep learning. And then we moved on to doing self-supervised image denoising, where you never have to see any clean data. The clean data is predicted, and you train only on noisy data by subtracting only the pixel noises that are on the images. And we did that also for cryotomography and for other data modalities. So here, every time you see something very contrasted, it is the result of our predictions, and when it is not contrasted, it is what comes from the microscope. So obviously these tools are useful and are enabled by deep learning technology, but this is not what this talk is about. I have one more example that is really fresh off the press. Some things are still in press. You know that if you do fluorescence imaging, the number of fluorophores you can simultaneously image, or a multiplexed image, is very limited. So if you would acquire an image where you label two structures with the same fluorophore, and you could later tell these two superimposed parts of your image apart with the power of deep learning, we think there is some value to it, and you can see that it works. And this is Assaf's project in the lab. This all just to introduce what we are doing. Now I switch gears. We do method research, and other people do that too. And we come from the computer vision and machine learning areas, and for various reasons, we now work on life science data. And these methods will be cast into tools that then will be used, as we saw also in the previous two presentations, by life scientists in the context of bioimage analysis. And here it's like a beautiful life cycle that I hope will become more commonplace in the near future, where then the users of said tools will upload raw data, metadata, maybe even labels that are generated by the various tools Anna told us about, so that they will be put into infrastructure, shared infrastructure, from where we as method researcher can take them out again and create better methods. And then the cycle can begin again. Important here to point out is that this method to tool transition is a job for research software engineers. These are highly skilled people that are not spoken about and not like valued enough in this world at the moment. So I try to kind of do that by singing the ode to these wonderful people by, for example, writing this opinion piece last year, or this opinion piece, which I invite you to read up on, but if you don't, that is fine too. What I want to come to now is, even if this life cycle and development cycle is in place, we also heard about fairness, right? Everything we do, and many groups do nowadays, might be open source. So the method might be open, even some tools might become open and are somewhat usable. We heard about zero cost today already, and this is what we did 2018 for this care denoising approach. So the usability, to some degree, we can checkmark that. It's not as easy to use as we would like, but I think we do an okay job. Reusability already is much less clear. Of course, what we do, you can now reuse because we put it out in ways that are enabling this. But once you use it, will you yourself make it reusable for yet others? That depends very much on how much time you have and what your attitude is. So we had this idea a number of years back where we said, maybe we should build an infrastructure, the Bioimage Model Zoo that you can find on bioimage.io. We did this with many collaborators over the years. That is from its inception fair. Every model in the Model Zoo can easily be found. And then it can also be accessed and downloaded and used in different ways, in different tools. So it's interoperable. And of course, therefore, everything that is up there is also reusable. So AI for Life is an EU-funded project, an infrastructure project that enables us to kind of come together, a number of teams, 10 total groups, to kind of build this infrastructure. So what is this infrastructure? First of all, the intention is that we bring the life science community and the computer science community closer together by helping us to devise and develop AI methods, but then cast them via standards we defined into a representation that lives one layer of abstraction above the standard tools that exist today, so that we can also go with the time and still have these models in the Model Zoo according to these standards in a fair and reusable way. So we cast these models into these standards so that these methods make it into a zoo and make it reusable and interoperable via different open source software tools. So if you go to Fiji, or if you go to Elastic, or if you go to various different tools, you can just give a unique identifier of one of these models. It will be downloaded from the model zoo. It will run on your data in the tool of your choice. We are not quite there yet. I think we have maybe 7 supporting tools, and they don't support 100% of the models. But AI for Life is only 1.5 years old, and there's still 1.5 years to go before this project ends, and then something else will come and continue this work, at least I hope. There's 2 more things I would like to point out. We also do open calls and challenges. And now let me quickly go through all of this. It was clear from the beginning there will be no time for a live demo, so you have to live with a pre-recorded video. If you go to Bioimage.io and you click on any of these model cards, a model card will open. You can browse through it. You see who uploaded it. You know, credit is given where credit is due. And then you can click on test model. What happens next is that an image.j that is cross-compiled for JavaScript is opening in your browser. You can drag and drop your favorite image, or you can click on load sample image. An image will open, and then you can click on run model. Now some magic will happen, and some working computer that we kind of pay for at the moment will run this data through this model zoo model and give you the output. And then you could say file, save as, and save it to your disk. So a lot of the infrastructure is already there. The next step for us is to see how to scale that and to bring it to people in a more useful and more at higher throughput, at higher accessibility. The open calls, and Vera will join us later, so you can ask her many questions. And I will also say a few more words in the next section of this talk. The open calls are possibilities for anyone who wants to apply that we help them solve their analysis problems. Similar to how Anna described that the SciLife Lab is giving this support to the Swedish community. We received in total 123 applications over 2 open calls, and there will be a 3rd open call by the end of this year. You will see that people come from different fields that I guess sounds all very familiar to people listening to this webinar, and they use tools that we as a community know. I will come back to the open calls. The very last thing I want to say now is that all the projects we took on, we describe also in a fair and accessible and reusable way openly on GitHub. So we describe what is the problem, what is the data, how do we install everything, because installation is difficult, as Beth pointed out. How do you use all your favorite tools to, at the end, have a segmentation and tracking pipeline, or whatever the original project was about. So I think this is one way to contribute to something meaningful to the community, because it's kind of a cut and paste follow tutorial that kind of can teach you how to do a thing as you should do it in 2023, 2024, with the tools that are available today, and for everybody to learn without that you require anybody else but an internet browser and a few hours of time. We also do challenges. So this is something that is more targeting the computer science community by engaging them in submitting their method research in the context of a certain task. So here we have a denoising task that we, on different data sets, that we kind of put openly out there, and then people can apply, submit their method, and this is currently ongoing, and then they can rank their... I'm giving a talk and you must leave. Thank you very much. So the value here is that we have at the end, we have a ranking of different methods, how they perform on different data sets. So if a cell biologist would like to know, oh, my data is similar to this one, what is the best performing method? You save yourself a lot of time trying different methods, because you can go there and get kind of informed prior on what you should potentially use. So this is all AI for Life utilities that we would like to bring to the community and give to the community. It's a very much a live project. I already told you that only half the time of the current funding period is over. So we hope that in the next 1.5 years, we bring many things over the finish line and become more accessible and useful. There is already a preprint for the bioimage model zoo, and there's a lot of other things that we also could contribute to. There is, for example, one thing that I really would like to point out, because I think that can be very useful, is a community-developed checklist for publishing images and image analysis. So it's really a very user-friendly checklist. It is not coming from AI for Life, but AI for Life people were involved in this work, and I just think it is very ASCB relevant. And then we also sit together more on a, like, let's say, policy level. There is at least 3 preprints, soon to be publications, where European and also worldwide we sit together and think, how can we create infrastructures that hold image data, that hold ground truth labels in an AI-ready way? If you remember my original life cycle, it's kind of all the way back. How can we enable people to store their data and store their metadata and store everything in an AI-ready way, so that we can kind of open the doors to a new generation of AI methods? Because data collection is very hard, and having the right data at hand is difficult. And all of this we do for this wonderful global community of bioimage analysts and cell biologists and developmental biologists. And AI for Life does it also for the European community. And I do it also for the Italian community, where the more we zoom in, the more we can say what has to be done, right? And in this institute that we are currently building in Milan, we are living this cycle as good as we possibly can. And then hopefully kind of make others interested to kind of jump on this bandwagon and join us in this global effort. I'm soon done. The second and third part will be not as long as the first, but I want to kind of repeat with my own words a few things that Beth and Anna have already pointed out before. I want to say a few more things about our experience in the open calls. So first of all, an open call looks something like this. So we say like, hey, I need deep learning help. I need somebody that kind of builds this missing piece, deep learning piece, that helps us to do our thing. And then we go over 72 applications and we do the best to select 8 that need some deep learning help, because this is what AI for Life should do, right? And then we struggle with different things, not with deep learning. We struggle with 3 projects having actually so little data that deep learning is anyway completely not possible. And one project didn't want to share their labels, although they said at application time that they are okay with it. And 5 projects were delayed because they were not actually ready to do the analysis yet. And the data transfer was such a pain that everything kind of, got delayed. From the 8 projects we took on last year, 4 projects were more or less, they only required us, it's a wonderful job and we like doing it, but it was not deep learning. Required us to kind of plug together, build a pipeline of existing tools. No deep learning, we didn't have to train any new models or adapt any existing methods. And only for half of them, some algorithmic development was needed, but it was also more classical than deep learning. Why do I tell you all this? That sounds weird, right? So first of all, this year we did a different job. We kind of advertised a bit different, and we hope that we find some people reading it. But the lesson learned here is deep learning is hard. And you have to ask yourself, are you here? Are you in the regime where actually doing either classical machine learning or even completely classical analysis workflows is giving you a better bang for the buck? Is this how you would say in the US? And many people are here, and you have to really look for the projects that really require deep learning so hard that it is worth investing the necessary time to do all the data crunching and development times and labeling and all the overhead. A good indication that you are maybe not there is if your distribution of how much time you put into curating your data is less than 80%, then maybe you are not there yet, right? So there is this white paper that shows that in a typical deep learning, and maybe that's a bit different in our community, but in a typical AI project, 80% of the time you kind of massage your data, you collect your data, you label your data, you do all the beautiful things that are so boring. And then 20% of the time is developing the AI solution. That was the optimistic end of the second part of my presentation. Let's swiftly move on and speak about methods, tools, users, and developers. So what are methods? Like, methods are this. Methods are hardcore, write papers that say, why is this method better than other methods? What is this method all about? It's not something that I think the people that listen to this presentation are, at least not the majority of you, will be interested in diving into a method at this level. A method is typically also, luckily the time progressed, this. Some GitHub repos that are more or less clean, more or less understandable, and run more or less. And if you are an expert, you clone this repo and you take care of all the dependency problems that you might possibly have, and then you find out how to run it, and then you can use it, right? And that is great. Maybe some small fraction of you are at the level where this is what you do, and then you should, and it's wonderful. But really, what most of you are looking for is a tool, and a tool is something different. A tool is, for example, one of the things we did some years ago is noise to void in Fiji. You open your data, you know how to do it in Fiji, you click on train and predict, maybe there's a few parameters you put in, and then the heavy lifting is done for you, right? And you just sit down and wait, most likely a long time, because in Fiji, chance is that you don't have GPU support, and already 4.5 hours later, you get a wonderfully denoised version of your data. Is it the most convenient? No. If I ask a research software engineer if they want to do noise to void in this way, the answer will be no. But for people that have 100% of the day filled with sample preparation, going to the microscope and doing biology, this is wonderful. This is accessible. You don't need to go and do courses first. This is Fiji, and Fiji is maybe old because now we all want to do Napari or Dubi. So we have the same thing also done in Napari. Napari is a Python-based viewer. It's wonderful. CZI did really a fantastic job in funding all the work that was required to make this happen. And at some point, people were asked to put more and more plugins into it, and lots of functionality you can find also in Napari, and that is wonderful too. But tools can also look differently. We saw this very easy drag and drop website from CellProfiler. What a nice tool. You drag something in there, you get the result. It is good, great. It is not good, bad. But it's easy to use, right? A tool. Zero cost deep learning for microscopy. You can even train your own CellProfiler model. It is maybe not as easy to use as dragging and dropping, but it comes with a lot of extra efforts to make the use easy. And then we also heard about the CellProfiler UI, if you manage to install it. And you can find various places in the internet that try to make this installation simple. Spoiler, it is not. But once it runs, it is relatively straightforward. So, in order to make methods to become tools, we obviously need users that want to use the tools, but we also need developers to create these tools. And I spoke about it in the context of AI for Life, and I spoke about it in the context of this beautiful life cycle of data and methods and tools. I think it would be great to do that, and I stole a slide from Beth because I went 10 minutes before this whole thing started and snuck it away from our shared slide deck. This is the place that I can just reiterate you should go to and ask for help. Many of the developers and many of the power users and many of other users are on the forum, and they will see your question and they will try to answer as good as they can. So this is all I wanted to say, but since I promised I want to go over time and I'm still in time at the moment, I will spend another 25 seconds for 2 more weird summary slides. If you have data to analyze, go to your core facility. Let's hope you have one. Otherwise, ask a friend if they have one and snuck in and get some expert that helps you. Or find a suitable collaboration. Or visit one of many courses. There are many courses in the US, in Europe, and all around the world. And that's for sure a good way to kind of get into the analysis mindset. And then ask yourself, what do you want to be? If you want to be a user of deep learning tools, great, see above, go to courses, have collaborations, go to a core facility. If you want to be a developer of deep learning methods, or if you want to be a research software engineer that kind of makes tools out of methods, welcome, colleague. Please get in touch with any of us that spoke today. Join deep learning courses. We have them at the MBL in Milan, in Heidelberg, lots of stuff. Just use Google, and we will be very happy to welcome you for 5 days, for 2 weeks, and teach you everything, share with you everything we know. And this is how I'm 1 minute over, and I stop talking.
Yay, well, thank you, Florian, not late at all. So next up, we have a panel discussion on really the practicalities of analyzing microscopy images. And we have 2 new panelists. So I want to start by letting them introduce themselves. Vera, yeah.
Hi, hello. Oh, sorry. Okay, so I'm Vera. I'm a part of Florian's group here in Human Technopole in Milan. I am from the industry background. So in the previous years, I worked in several deep learning startups, mainly applying deep learning to computer vision problems. I would describe myself as a deep learning engineer, so I write code to help people deploy deep learning methods and also research them myself. And a year ago, I joined Florian's group and got my first taste of bioimage analysis and start using Fiji and CellPose and all these wonderful tools. Yeah, so that's all I wanted to say. Thank you.
Okay.
Hi, Mr. Benjamin. I'm a postdoc in Beth Cimini's team. I'm a cell biologist, and I joined Cimini's team last year and trying to understand how the different image analysis tools that were mentioned during the webinar today can be used for microscopy images and understand them better. And I've used like CellPose and other tools that were discussed today.
Okay, so first question, let's ask Beth, and we've already talked about this multiple times, but I want to ask it again. So imagine you're an experimentalist in a lab and you have a bunch of data that you want to analyze. Say you want to segment cells. What do you do? What do you do first?
It's a great question. Hopefully, again, as Florian and Anna alluded to, you have somebody who's done this before that you can ask, because often it's really easiest to just sort of talk to somebody. Anna pointed out the Global Bioimage Analyst registry, where you can find someone who's near you or who has expertise in sort of things that you know about and care about. Or again, this image.sc forum is a great place to go. But it was really great to from Florian's talk to think about, again, how much data do you have? And it's also good to think about how accurate does whatever you're trying to analyze need to be. Do you just need to count cells? Then you might actually be able to do something that's pretty simple and doesn't need to be, for example, super accurate at finding cell boundaries. Understanding what is the biology you want to learn can actually then sometimes lead you to the right tool, because it's going to tell you what are the hard parts of your analysis and what are the easier parts.
Does anyone else want to answer as well? If not, we'll keep going. What do you do?
So
yeah, go.
So I mean, this deep learning magic is fantastic, right? But how do you know if something doesn't work as expected? I mean, how do you know that something failed or give you a completely crap answer?
Mm-hmm. Yeah, it's really important, whether you're doing deep learning, classical machine learning, other kinds of classical image analysis, to build in some way where you're doing sanity checking and you're checking to make sure that everything is giving you the answer that you think that you're getting. So for finding objects, we often recommend people overlay outlines of the objects that were identified. Or if you were doing classification problems, sort of some sort of a picture with, you know, what are the classes? Building in controls so that you can make sure that, you know, by looking at the quantitative numbers, oh, in my negative control, it shows very little of my, you know, marker, and my positive control, it shows lots of it. So human sanity checking is still, for my money, the best way to do this, because you are the person who understands your biology, and you are the person who's going to know if there's a problem. So building in ways that you don't have to just take deep learning's word for it, but you can actually go in and evaluate yourself if it's doing the right thing is so, so critical, no matter what kind of analysis you're doing.
I also just want to add, using deep learning doesn't mean to lose control over validation, right? The novelty is not that something magic happens and the result drops out and you don't ask questions. The novelty is that the results that drop out can be of higher quality than with other methods. Not even in all cases, but in many cases. Segmentation is a good example, right? Segmentation clearly made a jump with the advent of deep learning methods. But that doesn't mean that you shouldn't do all the necessary checks and controls. You should be as suspicious as possible. That is kind of part of being of your job of being a scientist, right?
So I'll follow up on that. And so, I mean, what are computational controls that you can use, right? I mean, there are experimental controls that you can check, but what would be experimental computational controls that you can use to validate your method? For example, classification, I don't know, or anything else.
I think Beth had a few things. I just unmuted and answered. This is how I operate, it seems. If you let a few of your favorite or randomly selected humans outline your objects in the context of segmentation, first you can compare to the same objects that were automatically segmented and labeled by your friends, but you can also see what is the inter-observer distance. And if your automated results are anywhere nearby, that is a good indication. If I would review something and I see, you know, also only on a small subset of the data that, but maybe statistically significant, this control was conducted, how should I not be okay with this way of proceeding, right?
I can maybe add that there was this very beautiful publication with metrics reloaded. I can maybe post it where you can explore a bit which metrics there are to analyze how well your outcome fits. And there's even some interface where you can click together and find the correct metrics.
This is a really, really valuable, I don't know, 200-page preprint or something crazy, right? But you are not supposed to read it from top to bottom. They have a website that really guides you to what are the right combination of metrics for your use case. And spoiler, the same people are now also working on because it's all very detection and segmentation heavy. They also work on other tasks and metrics in the context of other tasks. So I think that's very valuable.
Okay, now we have a question for the audience. Maybe we'll start with Anna. It says, this is from Claudia Vasquez. I'm very new to deep learning. I've only started dipping my toes into it. So thank you to all the speakers and panelists. I have 2 questions. What defines a small data set versus a large data set? For example, how do you know you have enough data for training, or is it just trial and error? And 2, is it better to train on raw or denoised data? So what defines a small versus a large data set, and is it better to train on raw or denoised data?
I guess the size, I don't know. How do we do that? So we usually, it's a bit more feeling of if it's more like, guess the deep learning projects that we had was indeed more like thousands of annotations that were used. And ideally, I guess you can, of course, you can try it out and see how many annotations you have, and then you look at kind of the accuracies you get while training and how it works and avoiding overfitting. For me, it's always a bit difficult to say in advance, how much data you need. And then, but typically it's, I don't know. In what we had so far, it was more indeed going into these thousands. But then, Beth, you were also mentioning already kind of starting not from scratch, but starting from a pre-trained network, and then you need much less. And that, indeed is the case. So it depends a bit. And then the denoising versus not, I'm actually not completely sure. Can I pass this on to the care expert? I don't know, I have a guess, but I want to hear the expert opinion.
Unmuting helps. Denoise before was a good question. I really liked it. So in the past, so there is papers that we wrote and others wrote, and it can be very helpful to denoise and then use the denoise data to do segmentation on top. There is also papers that do that together, and the latest iteration of this is actually the CellProfiler paper, where they show that doing different kinds of image restoration before the segmentation is actually very helpful. So ideally, this is done by the method, or let's say by the tool you use, that is encapsulating a method that you can read about, like CellProfiler. But if your tool doesn't do that, it can absolutely help. There is evidence and there are papers written that show that even doing it sequentially, first denoising your data and then doing certain analysis on top, deep learning analysis on top can be helpful. For sure, from the very inception of the idea of doing denoising and then doing any classical analysis is the way to go. And for some things, you have to go back to the noisy data, but maybe that's the answer to a different question.
I'll just hop in with an answer on the big versus small. It really, really depends on if you're fine-tuning a model that somebody else made or if you're training from scratch. I think the figure that I showed from the CellProfiler paper, which if folks want to refer back to that, they can. It took them thousands of patches of trained cells to get the correct answer from scratch. And with taking a pre-trained model and just having a human fix images one at a time, it took 5 images. Which, an image is composed of many patches, but still it took far less data. It's really hard to know if you have enough data if you're not familiar with how these models work. So again, this is where I'd really say if you're a complete beginner, talk to an expert. If you're somebody who just wants to get started with deep learning, you can certainly try it and just sort of try with however much data that you have. But it's going to be really important to hold out some of your data in terms of validation data and say, I'm not going to train on this data. I'm going to train the best model I can, and then I'm going to see how it performs on data it's never seen before. And you'll get a sense very quickly of how much overfitting has happened, and therefore how much more data you need to train a model that's sort of reusable. It also, the last thing I'll just sort of say on that is it really depends why you want to do deep learning. If you want to do deep learning to make a deep learning model that you want to then reuse and share with other people, you're going to need a lot of data because it's going to be used in a lot of contexts, and it needs to be very forgiving of a lot of different, say, imaging conditions and batch effects and things like that. If all you care about is sort of getting the right answer, if deep learning is just sort of a method to get you where you want to go, you don't care if you overfit to your whole data set as long as everything is correct and you've human verified that everything is correct. So a little bit is going to depend on your goals also.
Okay, so I have a question to Saganya. You did your transition, right, from biologist to computational biologist recently.
Yeah.
So maybe you can tell us a little bit about your experience, what worked, what was hard, and how did you do it? How is it possible?
Yes, as with the biologist, the main question began why I need computational experience, because I was struggling to analyze my intermediate filaments of cytoskeletal images. And that's when I realized I should gain more experience in the computational side which will help me analyze. To begin with computational knowledge, I attended a Python boot camp in the university, and also There are like, I would recommend like very good notebooks by Peter Bankhead and also Robert Hesse on using Python for bioimage analysis. I'll also post the links in the chat. Then there are like a lot of courses available online, like on YouTube as well. Like if you want to focus specifically on deep learning, there are tutorials available, the recorded tutorials from Images to Knowledge, which helped me a lot too. And I also took the deep learning course in MBL, and there are other courses like for deep learning from Human Technopole, as Florian mentioned as well. And there are other courses, which was really helpful for me to understand what these image analysis tools are and how I can use it for my analysis. And finally, if you like the image analysis so much and want to make it as a career, I would recommend joining the postdoc program by Beth. And yeah, being a part of it, I'm learning even more. Yeah, I'll also post these links here, the different resources that I mentioned.
Okay, so where is my next question? Oh, here it is. So any thoughts about, is it going to be any easier with the large language models that are going to be around? I mean, is this transition and using these tools is going to be simpler or more, right? More easy?
I can say something. There is very recently a number of like chatbots and other kind of tools. Robert Haase has one in AFLive Way, has one semi-accepted somewhere, and Loïc Royer from the CZI, I think, has a really nice Omega model out there. So the hope is that this can help, but it is a bit too early to really assess what this help will exactly be. I can only say what kind of the promise is. The promise is that you can ChatGPT style describe your image analysis problem, maybe even upload a small example image, and what you get back is a feature script that does what you want. And that will not work 100% in all cases, but it will most likely be better and better tailored to your concrete problem than the next best thing you can find in the internet on Stack Overflow or so, right? The problem there is that something that brings you, let's be positive, like 95% to your solution. Who will point out the 5% that it does do slightly weird with respect to what you intended to do? And is this 5% mismatch now a problem for you? Or can you live with it, right? All completely unclear. I think in 2 years we will know more. And it is certainly a good resource if your intention is, I want to become more proficient in writing my own little scripts. For sure that helps. Then you will anyway go line by line through the script and still try to understand. It will show you things that otherwise would be hard to find. So there is absolutely value to it. But I have to still remain with my finger stretched to the sky and say, but let's wait a bit. There will also, something will not work as great as we hope, and we have to still figure it out.
I think I can also imagine that it helps when you learn the beginning. So if you're indeed a biologist that now want to go into deep learning, and then exactly the kind of these questions like, how should I split my data set for training and validation and so on? And I think for these things, it could be helpful to ask a chatbot instead of Google maybe, and I mean, to get a bit kind of accumulated knowledge then. Yeah, so I think for this kind of learning of things that are around, it could be useful. And then, yeah, for the more specific, there's still some evaluation to go.
I think one issue is also that in order for bioimage analysis chatbots to be equally amazing as what we are now maybe used to from GPT-4, I'm not 100% sure if we have enough data already accumulated in such ways that we can actually train something that is not very biased in what solution it will give you, or will do obvious mistakes. So I think we have to run a few years through the method, tool, data cycle before we have the ability to really, open the doors, as I said in my talk, open the doors to the next generation of AI tools for bioimage analysis.
Can I add something? I had very success with just chatting with ChatGPT about biological problems. And I had one success story where I had a analysis problem that I couldn't solve myself, and I just went back and forth with ChatGPT. It didn't give me an exact solution, but it pointed me in the right enough way from where I could infer the pipeline that I needed in the first place.
Yeah.
So I would really suggest that if you encounter some kind of hard problems, to try at least to use it as like a body of sorts to run your problem against, that maybe you can find an answer.
Mm-hmm. Yeah, that now inspires me to also say one sentence more. I get more emails, applications, GPTs that are obviously, some parts are not like you curated anymore. And I think what Vera just said is absolutely right. If you have a learning group in university, right? 2 or 3 people come together, all 3 of them have no clue what they will know in a week from now when they've learned to study together for a week, and still they are more efficient than if anybody would do it on their own. What's going on, right? Exactly what Vera explained is going on. ChatGPT should not be seen as like the oracle that will give you the 100% solution, but it will come with things that you would not come up with yourself. And if you kind of see it as a partner that you can ping ideas against, and that gives you kind of some indications and pointers, you can progress faster than alone. And this is a mode of operation to use GPT in general. If you write a text, or if you need an analysis pipeline, or if you just need to understand something about biology, that I think is absolutely valuable and valid, right? But a person that then takes a response and cut and pastes it into the introduction section of the thesis, not a good idea, right? Instant fired. It's not the way a scientist should operate, right? You take it as entropy, as something that maybe gives you a few bits more than you can generate in this moment yourself, but then you have to kind of go over it again and think and be a responsible scientist.
Okay, great. So I have another general question. So I don't have a bioimage analyst in my institute. So I'm in my university. So I'm going to the next building and asking the computer scientist, right? Knock on his door or her door and ask for a solution. What do you think about this versus approaching a bioimage analyst?
I think a lot depends on your question. If you want to know what's the newest, coolest thing in deep learning, go talk to your computer scientist friends. What bioimage analysts tend to be good at is sort of understanding the intersection of the biology and the computer science. And so if you need to know what is the best model for this particular biology, a bioimage analyst may have had more direct experience with that. It's a whole spectrum, and there's lots of people who would place themselves in various things between computer scientist and bioimage analyst and biologist. But it depends if you want to know sort of theory or application, in my mind.
I think as an international community, we have to ask ourselves the question what we want, right? Do we want that analysis are done like they were done 10 years ago, just with a few more tools that kind of your lab, Assaf, grinds out, or maybe labs like mine and many other labs in the world, right? Or do we want to give better support to kind of improve the utility of acquired data? Data is not as cheap anymore as it was once, right? And now any old microscope costs $500,000. So investing also in the infrastructure, also human infrastructure, to make the best out of this acquired data, of this invested money, I think is important. So if there is an institute where a lot of great data is imaged with rather expensive, machinery, but also like just the wet protocols will cost a ton of money. I think it is not too much to ask to have some image analysis support on campus. Now, I think it's totally valuable if you don't have that to go to computer scientists, but you have to understand that they have a life as well, right? They need to survive, they need to write their thesis, they have habilitation, get the PI position. They will not have the time to do tasks that are not like salvageable as papers for themselves. And many of these problems are not. And this is why bioimage analysts exist and why research software engineers exist. And maybe then you have to team up, show some success, some publications that are better than what you would have done alone and whatever, and then go to your dean, to your I don't know, the political deciders in your community and tell them that look how the world does it, look how Broad does it, look how new institutes do it, how EMBL does it, how EMBO does it. Maybe it's time to just hire 1 or 2 people. And in 5 years, we will see that it was a good investment. This is my prediction. And yeah, depending on where you are, this will be more or less possible, but I see no other way to go into a future where we make better use of our data than this.
Okay. On that note, our panel discussion today is done. We hoped it was a worthwhile session for the audience. I think it was exciting for us. Our next session is on July 9th, and it features tandem talks and other talks by experimentalists, theory pairs. Right. So we hope you'll join us for the next talk, and thank you to all of our panelists today.